Hi, everyone. Welcome to STEM From's podcast, Where Does Your Journey Stem From? Hosted by myself, Dr. Karina Minardi. Today, we are joined by a creative scientist and guest, Mai, who is currently a graduate student at Brown University. Let's welcome to the stage, Mai. Hey, Mai, how's it going?
Hello. Hi, everyone. It's going good. How are you?
I am doing well. We're excited to have you. Mai Huynh is a fourth-year PhD student in Pathobiology program at Brown University. Her research in the Jameson Lab revolves around the complex relationship between the microbiome and the immune system in the context of cancer. She is very passionate about a breadth of topics, ranging from music theory to political science. I'm excited to get into this. She has been involved in social justice and equity initiatives since very young and hopes to pursue a career promoting diversity in all types of research in order to make healthcare more inclusive. That was a great mission, Mai. So let's start off a little bit about you telling us not only about yourself, but also your background.
My name is Mai, like Dr. Minardi mentioned. I'm a Vietnamese and Pacific Islander. So that combination has been a barrier, I guess, in terms of making a space for myself in the scientific field, just because there's still quite an underrepresentation of Southeast Asians and Pacific Islanders in science. And so that was kind of what allowed me to see all of the gaps in the way that science makes space for minorities, as well as how it caters to minorities in its practice. So I know it might sound cliche, but one of my biggest motivators is helping other people. One of my closest friends in undergrad used to say, always put people first, as in being there not only for yourself, but for people around you and advocating for those in need. So,
No, that's great. I think, and while it is, I guess, quasi cliche, I think to have a mission in which you actually tie something to tie what you do on a daily basis to something as important as that, as helping people, I think is really critical. I think that also lends itself to, the inclusivity of healthcare to the bio point. And so that drives you as a person. How did you get into STEM, pathobiology, an undergraduate degree? Tell us your story there.
sure. So, oddly enough, when I graduated from high school, I actually really wanted to go to music school. But my parents were very against the idea for a lot of reasons, and also by virtue of the fact that the only music schools that I got into were out of state, and I lived in Florida when I was younger. And we just simply weren't able to afford to go to I couldn't afford to go to a school out of state. So I went to an in-state school, and my parents were trying to move me in the direction of STEM. And my sister was already majoring in a STEM field at the time. But because it wasn't something at the time that particularly interested me, I kind of just arbitrarily chose a STEM major, which I know sounds strange because, it's like your college major, but, at 18, and I was kind of like, I wanted to go to music school. I just was like, oh, this will be fine, whatever. And I landed on microbiology and immunology. And it was really in the introductory class of that major that I realized how fascinating and complex biological systems are. And I realized how many different facets play with each other and against each other, which is, I feel like, not unsimilar to the way that music is composed, but also like in a lot of I feel like our STEM is a lot more artistic than people realize. And yeah, and I just I ended up really enjoying it and enjoying learning about the field. So yeah, that's kind of how I ended up in STEM, and I ended up really loving it.
So, what musical instruments do you play?
When I was applying for school, I was primarily playing the piano, and I auditioned with the piano, but I also played the violin and viola, as well as the guitar and ukulele.
Wow, that's amazing.
Me too.
We had a previous guest who played the French horn, and I asked him a question around creativity and music and how that has actually influenced his STEM career and his research. So I ask you the same question. How has music and creativity in that realm influenced you?
Yeah, I feel like you know, nothing is perfect, and the in practice also nothing is perfect. So when you play a song, for instance, like of course there's notes on the sheets, but especially a lot of more difficult songs, they have a lot of space for improvisation, and I feel like in science it's a lot of improvising, like just like being on your feet when it comes to making a decision, especially when something doesn't turn out immediately the way that you expect it to, or trying to find a way to make things work, which may not always seem like it's supposed to on paper, but sometimes the way doing things in a kind of unique and creative way helps you get the results that you're looking for, which might not be immediately intuitive.
Well, that's great. I think that that's precisely right. And it causes you also to think a little bit outside of the box, too, which I think is a definitely a skill to have in STEM. So you talked a little bit about immunobiology, microbiology, your introduction there. Is that how you got into pathobiology? Did you do research as an undergraduate? Tell us a little bit about that.
Sure. So actually, as an undergraduate, the research that I was involved in was not immunology or microbiology really oriented at all. It was all cancer biology, which is more of, I suppose, like the molecular biology of cancer. Um, and But yeah, I mean, I found the research very interesting. I was primarily working on redox protective proteins, which were proteins that kind of help to protect against DNA damage in our normal cells, but that these proteins tend to be taken advantage of in cancer systems to kind of allow the cancer cells will manipulate these proteins to kind of create the kind of mutations that they desire to function in the way that they want. And so, in kind of dysregulating the system, we're essentially kind of like where cancer cells are already like having their foot on the accelerator when it comes to cellular division. It's kind of like really slamming that accelerator down, which forces the cancer cells to hypothetically crash. So this is kind of the system we were thinking of. I think it's called synthetic lethality. Um, but but yeah, I found it very interesting, particularly because cancer affects so many people, and there were so many people that I was working with, as well as the people around me who are affected by cancer. So I wanted to see firsthand what that research looked like, and how that research actually results translationally to helping people. So I really enjoyed that research as an undergrad. And then pathobiology, I feel like is kind of like a pretty unique term that Brown has, but it ultimately, I feel like, encompasses microbiology and immunology in that, like looking at pathogens and how our bodies respond to those pathogens. Um, so I felt it kind of went along the line of what my major was like, although it didn't really match up with exactly what I did with my undergraduate research. I felt that I really wanted to explore this field of science, which I had for the past essentially 6 years before I started graduate school, because I spent 2 years after undergrad working as a technician in cancer genetics lab. Um, and so I really wanted to kind of look more into this field that I hadn't really dove into before, which is more of an immunological aspect. And yeah, I felt like it was very rewarding. I'm learning a lot in the program.
You talked a little bit about taking a couple years off in between undergrad and grad. Can you talk a little bit more about that? Why? and what it gave you sort of for graduate school in preparation.
Honestly, I felt like those 2 years that I took off before graduate school were really, really valuable to my experience and to my career trajectory. I don't think I would have been able to navigate graduate school in the way that I currently have if I hadn't taken those 2 years off. First, I feel like just spending 2 years where you're not really doing anything except bench work really helps to put you in the mindset of this is what my day to day life might look like, and will I actually enjoy this? After I graduated from college, I honestly felt kind of indecisive on how I wanted to um, uh, pursue my career goals, which I think ultimately I knew that I wanted to do something where I would be helping people, but I didn't know how and what, like at what angle and what facet. So I think doing those 2 years working as a technician really helped me to see, like, okay, is graduate school going to work for me? Is it going to be something that I can see myself doing? And yeah, so in those 2 years, I felt like I became more familiar with that bench work and could see myself doing it for a couple of years. And it also gives you a lot more time to just think about like your career because sometimes like you're just on the spot all of a sudden like in your last 2 years of college trying to figure out what you want to do, and I think it's a little bit too hasty. So, and you're also just, you're busy with classes and like extracurricular activities and all these other things. It's really kind of hard to focus only on your career, and having those 2 years also helped me think about what I want to do a lot better.
No, I totally appreciate that answer. I took it's funny to juxtapose, I think, your approach to my approach. So I also took a year off in between undergrad and grad. And what I did was I didn't do any research. And I missed it and I started missing it. And I went, oh, if I miss it, that's actually a good thing. So it's interesting, to kind of take it from those two perspectives, but also to do some reflection and to do some self reflection. I don't think there is enough time during undergraduate to really think about long term career trajectory or what you want to do, especially if you go into graduate school. So I very much appreciate your answer, but also say, like it depends on the person too, which
Yeah.
I appreciate. So you talked a little bit about career goals, and you talked in the bio. Your goal is to make healthcare more inclusive. So I'm curious from a sort of a large overview perspective. What do you want to do when you grow up?
Yeah, so that's a sometimes I'm like I'm already a grown up, but like at the same time I still feel like such a kid, especially like surrounded by all these academics and stuff. But yeah, in my mind, my current goal is I'm interested in working for like a nonprofit organization, maybe being involved in like lobbying for certain policies to promote equity, that kind of thing. But I'm primarily interested in maybe getting involved in the United Nations or the World Health Organization. Um, I've also always been like a huge feminist, so I'm kind of interested in maybe moving in a direction of like reproductive health and gender equity. So yeah, those are kind of what I'm leaning towards.
No, that's great. How do you think having a background in STEM will help you in nonprofit, political sector, policy, advocacy? I'm curious about what you think are translatable skills.
I think there's so many translatable skills when it comes to graduate school. He was saying that graduate school in and of itself counts as experience. It counts as work experience because you're not only working, but you're also a student. And in that time, you're involved in, delegating your project. You're also involved in managing the different projects, because usually there's more than one project that you're trying to juggle at the same time, and multitasking, and not only the bench skills but also the kind of mindset that you need to have in you know working through issues and troubleshooting those problems that you're experiencing at the bench, I feel like are very valuable skills that I think all employers appreciate. So I feel like getting a PhD in STEM, but just a PhD in general, really gives you a ton of experience and skill sets that would be valuable to any employer. So I think getting a degree there will help you succeed in any career that you would be interested in. And another thing too is I've been pursuing a master's degree in political science alongside my doctorate, which has been I feel like very helpful too, because I'm kind of seeing what political scientists think of science and technology, and also seeing the gaps that they don't quite understand about the way research is conducted. And I think it'll be really cool to apply my knowledge in both directions, because I feel also that a lot of scientists might be lacking in kind of that understanding of how politicians or diplomats make decisions around science policy. And I think it'll be kind of cool to transfer knowledge between those two fields.
No, I love that, and I love it for a variety of different reasons. One of which is that is sort of the mission of STEM from, right? Is to think about translatable skills where you consider a PhD as work experience, and there's a variety of different ways and reasons why. The other piece is that how I like to answer questions about scientists and broaching different industries is, as a scientist, you think about black and white, or you think about zeros and ones, and it's right or wrong. But when you actually think about other industries or other topics, it's very gray. The dichotomy maybe of saying political science plus scientists, there's not enough scientists in political science. There's not enough scientists, I think, pushing for advocacy and advocate groups. And so it's exciting that you're in that. So let me just say that. So let's actually talk a little bit about your current research. I'm excited to learn more. So if you could give us a little bit of an overview as to not only your research but the Jameson Lab and what the purpose is, I would love to learn more.
Sure. So I think the Jameson Lab broadly looks a lot at the lung, does a lot of lung-related research. Although there's also a major project in the lab that's also surrounding wound healing and how infection affects wound healing. But my project in the lab is specifically on the lung microbiome. And like I mentioned before, I'm studying this complex relationship between the microbiome and the immune system in the context of cancer. Specifically, I'm looking at the lung microbiome and lung cancer. And so essentially, we're trying to probe the composition of the lung microbiome in a mouse model throughout health, as well as the diseased lung, and how perturbations to the system affect not only the composition of the microbiome, but also, or the microbiota therein, but also how those perturbations affect the disease state of the animals that we're looking at, just so that we can get a better understanding of why, for instance, maybe therapies are, or lung cancer related therapies are not successful in some aspects, and if the microbiome plays any kind of role in the tumor microenvironment.
Or how would you talk about the translatability there?
That's a great question. So, in our minds, I think a lot of people might think that when we're looking at these microbiota, that they aren't relatable, like translatable from a mouse to a human. But I think it's more important, rather than focusing on the specific bacteria that are there, but more about the functions, like what are those bacteria doing. And a lot of bacteria, regardless of whether it's an us or an armadillo or a mouse or a bird, they share similar functions because they're there for similar reasons. And so in a tumor in a mouse, there might be bacteria that are near or promoted in the presence of a tumor. And that might be because of a very specific nutritional microenvironment that tumor is promoting. And so similar bacteria are going to respond similarly to specific nutritional states. So I think even if the bacteria themselves might not be as translatable, I think the about learning more about what they're doing there will still be very valuable information.
That's fascinating because I was thinking, what is the translatability, particularly with lung cancer? Um, so, so that's a great perspective, I think, from the Jameson lab. So, can you talk a little bit about sort of what your hypotheses are, what you're doing specifically, some of your experiments right now?
Definitely hypothesize that there are microbes that are present in the lung that might be promoting during cancer, that might be promoting a more regulatory state, that might be dampening some of the immune response to the site of the tumor. And so maybe changing the composition of the microbiome during cancer might change the way that the immune system is responding to the lung and help our immune systems find our tumors and respond to them, because it might be hypothetically that these tumors are cloaking themselves in kind of like a symbiotic bacteria cloak that might be preventing your immune system from seeing it as a problem, because your immune system is trained to not respond to bacteria in your body that are safe bacteria or symbiotic bacteria. And if these tumors are kind of cloaking themselves in these microbes that are considered safe to our body, it might be preventing our immune system from finding that tumor and essentially responding to it accordingly.
So, what does an experiment then look for you, or look like for you, I should say, with respect to, do you take out the tumor from the mouse model? Walk me through that a little bit more.
Sure. So currently, we're performing just a whole lung. We're essentially blending up a whole lung extracted from these mice. Um, at this time, we're still not at that stage where we're looking at like location specific extractions within the lung, because it is kind of a small system, and the lung is also a pretty low microbial biomass system. So it's a little bit difficult to extract bacterial genomic material from the lung. So we use whole lung extracts, just trying to see what the composition is looking like in the lung. Um, and so an experiment would be me exposing a mouse to some kind of perturbation, for instance. What will happen to the development of tumors if I expose these mice to antibiotics, for instance, intratracheally or intranasally, um, prior to the onset of a tumor. And so let's say I'll expose these mice to antibiotics, and then after a specific time period, we'll uh, uh, harvest these mice, harvest the lungs from these mice, kind of blend them up, and then perform a pretty straightforward microbial extraction from the sample. And then after we are able to isolate the microbes, we perform a DNA extraction from those microbes, and then we send that genomic material for sequencing. So then we send it to a company that performs sequencing for us, and then they give us back essentially a file that has all the sequences in it, and then we perform the kind of downstream analysis of, okay, what's in there, what sequences match to which microbes, etc.
So, how do you know, and this is going to be a fairly ignorant question, but how do you know if the mouse has lung cancer?
We use kind of two models. We use Cree-inducible mutation in one of the alleles of KRAS, which is one of the primary mutagens in non-small cell lung carcinoma. And so a lentiviral Cree recombinase, which is essentially we expose these mice to lentivirus that has a vector for Cree recombinase, will be intratracheally administered to these mice, and then it kind of acts like scissors and goes in and cuts at where when that allele has that mutation. So essentially, when it was without the Cree recombinase, it's essentially wild type. But once the Cree recombinase is exposed to these mice, that second allele in the KRAS becomes mutated. So then it becomes KRAS heterozygous mutated. And so that mutation is sufficient to induce cancer in these mice. But to ensure that they do have cancer, in half of the mice we extract, we treat a bunch of mice the same, and half of those mice we extract the lungs, and the other half we send for histology. And so we determine their cancer state by the histology.
That's so helpful. I was thinking, okay, do you do like a X-ray to actually look at the tumor state and stage of the mouse? I don't know. That's what I was thinking. But okay, yeah. Histology works too. That's obvious.
Yeah.
So what keeps you up at night with your research?
Yes, there's always this kind of back of your mind understanding in graduate school, like, okay, I only have 6 years here, 5.5 years here. Let me try and get this done as soon as possible so I can make this data as soon as possible so that I can write my thesis as soon as possible, etc. And because of the low microbial biomass nature of the lung microbiome, it takes quite a long time to perform those extractions, which is why it's still such an underdeveloped field in science, because it is so difficult to perform those extractions. And so sometimes I'm kept up at night by just the amount of time it takes to perform these experiments, and are they going to go anywhere, or is it actually going to mean anything? Am I actually going to find any meaningful results? Is this actually ever going to help anyone? But sometimes I think focusing on those things can kind of dampen your ability to perform in, day-to-day life. And also, it's important to focus on the here and now, and try and figure out what you can fix now and not focus on things that you ultimately might not have control over. So, yeah. I think as scientists, we're just so obsessed with control because, we have the capacity to do so in a laboratory setting. But I think for our mental health that a lot of scientists need to loosen our grip on our control of everything else in our lives, because the world is not a laboratory setting.
Well, and it's so funny too, because there's a common phrase I think in corporate culture that talks about progress, not perfection. And I think scientists, all too many times when you think about experimentation, it's okay, I got to get the right perfect experiment done, so I can get 100% yield, or as close as I possibly could to it, or whatever have you. And yeah, it's like progress. It's progress, and learning from your failures too. So we're wrapping up and we're nearing the end of our time. I do have a question regarding, though, if you were to interact with Mai 10 years ago, what kind of words of wisdom or encouragement would you have given to her?
Told her that, everything's going to be okay. I mean, I remember at that time being so worried and scared about my future, and thinking, since I had no idea what I wanted to do at that time, like, I remember being so anxious about it, and that would keep me up at night at that age, is thinking about that question. And, would it be worth it to take time off? And I would tell myself at that time, like, everything always works out the way it's supposed to. And, and sometimes people do need time to figure out what they want to do. And so don't hate yourself for that. Don't, stress too much about, taking the time to figure out who you are and what you want to do, because taking that time will really help you set the stage for the life that you're looking for. So, yeah.
Those are some true words of wisdom. I think the other piece to that is what you want to do maybe in 5 years is not what you're going to want to do in 20. So there's power in reflection, and there's power in what drives you as a human being. So that's really powerful. So thank you for that. And with that, thank you, Mai, for being a guest today on the podcast. You have an amazing story and some really fabulous research that you're pursuing. So I'm excited, and I think all of our listeners are also excited to kind of see your publications in the future and what it leads to. So thank you again. And for all of our listeners, don't forget to ask yourself, where does your journey stem from? Bye.
Thank you for having me.